Welcome. This is Richard Ransohoff. I'm Vice President and Senior Biogen Research Fellow for Neuroimmunology at Biogen in Cambridge, Massachusetts, and also Adjunct Professor of Molecular Medicine at the Cleveland Clinic Lerner College of Medicine. Let's begin with our learning objectives. What we'd like to do today is recognize the role of T-lymphocyte trafficking in immune responses. We're going to describe the immunobiology of multiple sclerosis and how lymphocyte trafficking is implicated in the pathogenesis of MS. We'll understand some strategies that are now used for targeting T-cell trafficking using both current and future agents. The current agents are natalizumab and fingolimod. And at the end, we're going to mention T-cell trafficking in other diseases such as inflammatory bowel disease, IBD, rheumatoid arthritis, and psoriasis. Let's take a look at the process by which lymphocytes move throughout the body and the reason that they move in the way that they do. Naive lymphocytes, which means lymphocytes that have not been triggered by recognizing a component of a pathogen, such as a bacteria or a virus, and these components are called antigens when they're recognized by either T cells or antibodies. Those naive lymphocytes, as it says at the upper right, enter the lymph nodes from the blood. So you might track a lymphocyte from the heart, traveling down through the body and entering what looks like a popliteal lymph node in the back of the leg. A lymph node is a way station where lymphocytes can experience material that comes from an infected patch of tissue, which drains its interstitial fluid up through the lymph into the lymph node. So at the bottom, you see antigens from sites of infection reach lymph nodes by lymphatics. And you can see that this individual has what looks like an infection on the maybe the medial side of the ankle, and that infected material is draining to the lymph node. We're going to talk about the encounter of the T cell with antigen and how that works later. But right now, let's just keep a focus on how the lymphocyte moves throughout the body. So it goes from the arterial blood through a high endothelial venule into the lymph node, searches for antigen, may or may not see it, and then if it does not, it will exit through the afferent lymph, ultimately to the thoracic duct and back into the blood. So that's the pattern of movement of naive lymphocytes throughout the body. They go from blood to lymph node to afferent lymph and back to blood. Now let's move on to a case history which we're going to use to understand the role of lymphocyte trafficking in human disease. A 32-year-old woman, a college professor, married with no children, but wants to start her family in 2012, has no significant past medical history, and she comes to you for consultation in July of 2011. In March 2011, 4 months before she saw you, she had optic neuritis in the left eye. Her primary care physician ordered an MRI brain scan, and while she was waiting, she had a spinal cord sensory syndrome lasting for 2 weeks. In your office, you perform a neurological exam which shows mild visual sensory deficit in the left eye corresponding to the residual of that optic neuritis, and her exam is otherwise normal. The blood work for anything that could mimic MS, such as B12 deficiency, is completely negative, and the MRI brain scan is consistent with MS, with one enhancing lesion. So you diagnose her as clinically and radiographically active relapsing remitting multiple sclerosis. You say that it's active because she's had 2 recent attacks, and you say it's both clinically and radiographically active because she has a lesion in the brain which doesn't correspond to any of her symptoms, telling you that the disease is still active at the subclinical level. You recommend that she begin a disease-modifying agent such as interferon beta, and after 6 to 12 months, you're going to monitor disease activity both by history exam and by MRI. After 6 months of interferon beta, as shown on the next slide, she has a number of new lesions. Compare the February 2011 image with that of February 2012, and you can see 3 arrows which point to 3 new lesions. And you can also see that the scans are pretty well matched for the level of section, showing that these are not being seen just because you're taking a different picture of the brain, but that these have actually popped up since you last saw her. Let's think about this science in the context of disease. Multiple sclerosis, an inflammatory disease of the central nervous system, is the best example of disease control by targeting leukocyte trafficking. MS has its onset really with quite a sharp peak around age 30. It's often said that it can be any time between even age 15 up to 50, but the majority of patients present in the 30-year age range, plus or minus a few years. 2 times as many women as men are affected. Worldwide, there's about 2.5 million cases, and the etiology involves components of genetic susceptibility and environmental exposure, much of which have been identified in recent years. Natural history studies of MS show that 50% of people are using canes or walkers for ambulation after 20 years. So this is a population of people, if you think about onset at age 30 and canes and walkers after 20 years, that's a bunch of people who are now 50 years old, having difficulty with mobility and at high risk to require use of a wheelchair before too long. MS became a treatable disease in 1993, and there are now at least 5 main classes of agents used for multiple sclerosis. Class number 1 would be the interferon beta products. Class number 2 would be glatiramer acetate. Class number 3 would be the monoclonal antibodies, natalizumab and alemtuzumab. Class number 4 would be the oral agents, fingolimod, dimethylfumarate, teriflunomide. And class number 5 would be the cytotoxic immunosuppressives, which, given the availability of the drugs with lower toxicity that I mentioned before, the cytotoxic immunosuppressives are rarely used anymore. In an individual patient, the efficacy of these agents ranges from excellent to negligible. Now, what I mean by that is that when you start a patient on a drug for MS, you really don't know what you're going to get, but it becomes clear within the first year or so of treatment that you either have quite good disease control or, unfortunately, in some cases, rather poor disease control. Overall, people do better with their MS treated than they used to in the natural history era before 1993. But what we don't know and very much need is biomarkers that would help us choose the right drug for the right patient. Because as in every other neurologic condition, time is brain, and we need to move towards the precision medicine era in MS when we can do some form of analysis that helps us choose the right drug for the patient when they first present. Getting back to leukocyte trafficking, three of these drug classes, natalizumab, fingolimod, and interferon beta, target leukocyte trafficking. In order to understand the role of leukocyte trafficking specifically in a disease of the central nervous system, we have to take a step back and consider the old concept. This concept is over 80 years old, of immune privilege of the central nervous system. What do we mean by immune privilege of the central nervous system? This slide illustrates experiments that depict what immune privilege of the central nervous system really is. In the right-hand panel, you see a graphic illustration of what happens when killed bacteria are injected carefully in a minimal volume into either the skin or the brain. If you look at the top line, you see the response in the skin. And what we mean by response here is the accumulation of inflammatory cells along the injection site. What you see is large numbers of cells accumulating, and that their presence at the injection site lasts for at least 6 weeks. It's an incredibly vigorous response. If you perform the same injection into the brain, the response is much, much less in magnitude. That's the lower line, and it burns out in about 4 weeks. So it's transient and wimpy by comparison to what you see in the skin. The left-hand panel just shows you how this is quantitated. This is a patch of brain tissue which was injected with killed bacteria and then studied 2 weeks later by immunostaining for inflammatory cells. And what you see is a very faint blush of inflammatory cells right along the needle track and nothing else. So this tells us two things. First of all, the afferent system by which the brain talks to the immune system is inefficient and slow. You've put something that represents what could be an infection directly into the brain, and the brain barely reacts. The implication is that if there is an adaptive immune reaction in the brain, meaning a reaction that involves T cells and B cells, therefore part of adaptive immunity, this reaction must have been initiated in the periphery. The brain cannot initiate its own T cell mediated immune reactions. That is the definition of immune privilege of the CNS. In order to understand how that works and why the CNS is so different, we need to look at a sort of generic immune response, and that's shown on the next slide. So we're going to look at adaptive immunity at the level of priming, response, and immune memory. At the upper left, you see a green box, and that's meant to represent a patch of healthy tissue. We might think of this perhaps as a bit of skin. Right in the middle of the image, you see what is called a dendritic cell. That's a specialized type of macrophage that lives in the tissue and simply waits there in case any pathogen should enter. At the upper right, now you see a red box around what is labeled inflamed tissue. And what this indicates is that there's been, in this case, it would be a breach in the skin and a pathogen has entered. The dendritic cell will essentially act like Paul Revere. It will take up the pathogen, engulf it because it is a type of macrophage. It will digest all the components of the pathogen and then turn around and display those components on its own surface for recognition by T cells. It will then use the afferent lymphatic, which you see right under the cell, and move into the lymph node that drains that bit of tissue. And within the lymph node, that dendritic cell will search for a T cell. Remember, we talked about T cells entering lymph nodes before. So T cells are regularly entering lymph nodes, looking for indications of trouble as presented by dendritic cells or other macrophages. The T cells do not use the afferent lymphatics to get in. They use the high endothelial venule, which is specialized type of endothelium that tells the T cells that this is a place from which they can enter a lymph node. And there are only two types of T cells that enter lymph nodes: the naive T cell, which has never been triggered by antigen, and the central memory cell, which we'll talk about later, and keeps track of whether a pathogen that the organism has seen before has re-entered the scene. If you look below the lymph node, you see the antigen-presenting cell with the antigen mounted on some surface structures, presenting that antigen to the T cell. And in this case, the T cell recognizes the antigen as being foreign and corresponding to what can be bound by its own T cell receptor. And the T cell will then undergo a very rapid process of what's called clonal expansion, but is simply multiple cell divisions, which give rise to daughter cells that recognize the same antigen as the parental T cell that first recognized it, as mounted on the surface of the dendritic cell. These rounds of cell division give rise to two categories of daughter cells. Some of them are short-lived effector cells, which will return to the infected tissue and orchestrate the clearance of that pathogen away from the tissue and initiate wound healing. The other type of T cell is the central memory T cell. Now that's got a blue box around it, and the central memory T cell is the one that can carry immune memory for years, sometimes for the life of the individual. That's the cellular basis, for example, of our ability to retain immunity to those pathogens for which we received childhood immunization. So central memory T cells alert the organism whenever a pathogen to which it's been previously been exposed has re-entered the scene. Let's take a more close-up look at what antigen-presenting cells tell the T cell. So the antigen-presenting cell membrane is shown at the upper part of this cartoon, and the T cell membrane is shown at the lower part. The T cell and the antigen-presenting cell exchange a great deal of information, most of it flowing from the antigen-presenting cell down to the T cell during an encounter that can last as long as an hour or more, which is quite a long time in the lifetime of a cell. So let's go to the left and look at the structures that connect the T cell and the antigen-presenting cell in sequence. So we're going to look now at what's labeled MHC class II and TCR. MHC class II is a molecule on the surface of the antigen-presenting cell, which the APC uses to bind antigen on its own surface, and that's the red ball, and show it to any passing T cell, looking for a T cell that has a specific receptor which can recognize that red ball, that antigen. And that is the first step in T cell activation. The whole process is called activation. But the initial process, or the initial step, is that by which the T cell receptor says, yes, this is an antigen that I recognize. And that begins the activation process. The T cell is then confronted with a fairly stark choice. It can either proceed then to die if it does not receive additional signals from the APC, or it can survive and begin to initiate proliferation. That second signal is given by what are called co-stimulators on the APC, and co-stimulators are represented here by molecules called B7-1 and B7-2, which were the first discovered co-stimulators, and they bind to a molecule called CD28 on the T cells. What this tells the T cell is that this antigen that you recognized is bad news and you're going to have to deal with it. The third set of signals are represented by all the cytokines that are listed on the right hand between the APC and the T cell. And it is the specific cytokines that the APC makes that lead to differentiation of the T cell. T cells have to be able to deal with a wide variety of pathogens, including intracellular bacteria and viruses, which replicate within cells, or extracellular bacteria, or fungi, or protozoal parasites. And a specific type of T cell response is most effective at clearing each one of those pathogens. The APC is responsible for deciphering the type of pathogen and giving the T cell a set of signals which tells the T cell what it is that has invaded the body. Is it a virus? Is it a fungus? Is it a bacteria? Is it intracellular or extracellular? So APCs don't make all of these cytokines. They make only the ones that are informative about the type of pathogen. IL-12, for example, for viruses and intracellular bacteria, IL-4 for protozoal parasites, and so forth. So let's get back now to the immune privilege of the CNS. What you see here is the hind end of a rodent, and you see a sort of very succinct depiction of what we just talked about as a generic immune response. A pathogen has entered the skin. The components of the pathogen will get into the lymph node, either in soluble form with the lymph or in the cellular associated form, the yellow arrow, associated with these dendritic cells. They will inform the T cells, and then the T cells will return to the skin and mount an immune response to clear the pathogen. Now, if we add in the head and the rest of the mouse, and you see the CNS label now on the left, we can see what's different in the comparison between the skin and, in fact, any other part of the body and the central nervous system. Specifically, the parenchymal tissues of the central nervous system, the actual spinal cord, brain, optic nerves, do not have any dendritic cells. There's no cell within the brain that will engulf a pathogen, leave the brain, and go to a lymph node to tell the rest of the immune system that something is wrong. That's the basis of immune privilege of the central nervous system. It's the absence of dendritic cells in the central nervous system. Now, we know that this immune privilege of the CNS can be defeated, as shown on the next slide. We know that it isn't absolute, and the reason we know that is because, first of all, when a virus enters the brain, ultimately there will be an immune reaction. We know that, for example, herpes encephalitis is a very inflammatory process. We also know that there are diseases in which inappropriate inflammation involving T cells, B cells, and the adaptive immune response affects the brain, and an example of that is multiple sclerosis. So how do we work around the immune privilege of the brain to get an immune response going there? The left-hand panel on this slide you've seen before. This is the wimpy transient immune response when you simply inject some killed bacteria into the brain tissue carefully without making a lot of damage and without disrupting the vasculature. And the second panel on the right shows the violent inflammatory reaction in the brain that results when you place the killed bacteria in the brain and then, somewhat later, immunize peripherally, meaning give an immunization in the skin of the same bacteria, and then come back and look at the brain 2 weeks later. First of all, you can see a large activation of the inflammatory cells of the brain parenchyma. And then if you look closely, you can see that the vessels in the tissue are, all of them, completely surrounded by inflammatory cells, probably in the process of entering the tissue. So we definitely can get around the immune privilege of the brain by immunizing peripherally. And it's remarkable if you think that what you've done here is put a small depot of bacterial antigen in the brain, then you've gone to the periphery and immunized in the skin. And essentially what you're telling the immune system is, there's something bad somewhere in the body, now go find it. And remarkably, the immune system did that quickly and robustly. So this gives us a lesson. The efferent system, by which the immune system finds something in the brain once it's been notified, is quite efficient. This means there must be a process of immune surveillance of the brain. So how does that work? Basically, what we're looking for is the central memory T cell of the brain. What is the central memory T cell that keeps track of what's going on in the brain, and how does it do that? Let's first look at the migration pattern of the central memory T cell. I told you before that the naive T cell, which you can see up at the top middle of this cartoon, uses high endothelial venules to get into lymph nodes. It will look for antigen, and if it doesn't find anything, meaning there's no infection going on, then it will return via the lymph, via the efferent lymph and ultimately thoracic duct, into the blood. So naive cells go blood, lymph node, lymph, blood. How about central memory cells? They have one additional stop. So they go from the blood to tissue. So they monitor tissues for anything that needs attention from the immune response, such as a pathogen. So blood to tissue, and then using lymphatics into a lymph node, and then eventually through the efferent lymph back to the blood. So it's blood, tissue, lymph node, blood. And that's the difference between central memory and naive T cells. So we're looking for a central memory T cell that could monitor the CNS. Some years ago, we began looking for those T cells, and eventually we found them in spinal fluid. The initial stages of the research involved doing analysis of cells in the spinal fluid of healthy people, and the reason we knew that they were healthy was that they had come to the Cleveland Clinic to get a spinal tap for some suspected condition, either in the emergency room or in the neurology department, and when we looked at their medical records, they were completely cleared of all concern about central nervous system disease. So we had a collection of spinal fluids that came from people who had something which eventually was ruled out. They had suspicion of something, and that was ruled out, and their spinal fluid was normal, and they were normal. When we looked at the cells in that spinal fluid, remember that you always get a count of 1 or 2 cells per microliter of spinal fluid. That means that there's 1 or 2,000 cells per mil, and since we have anywhere up to about 300 mils of spinal fluid in a person at any given time, this is several 100,000 cells in the spinal fluid. It turns out that these cells move in and out of the spinal fluid on a regular basis, and that they are virtually all T-lymphocytes. 3/4 of all those T-lymphocytes are CD4 T cells. The rest are CD8, and almost all of those cells, both CD4 and CD8, are central memory T cells. So we proposed a long time ago that these T cells carry out immune surveillance of the central nervous system. The person who did this research in my lab was Pia Kivasak. You can see her name at the bottom of the next slide that says T cells scan APCs in the spinal fluid of healthy mice. So after Pia made this discovery about T cells in spinal fluid and made the proposal that these are the T cells that carry out immune surveillance of the central nervous system, she wanted to go further and prove it. And in order to do so, she developed a technique to strip off the spinal meninges of healthy mice, stain the T cells green, stain the antigen-presenting cells red, mount the meninges in a solid matrix, which allowed her to do video imaging of these fluorescent cells, and made video movies of the cells interacting. And what I'm showing you here is a bunch of stills from one of her movies. What you see in the upper left is a T cell, which is round, approaching an antigen-presenting cell, which is much larger and is colored red. The T cell comes up to the antigen-presenting cell, extends a large, flat process, and begins to scan the surface of the antigen-presenting cell. What it's actually doing is passing its T cell receptor across the MHC molecules, the major histocompatibility complex molecules of the antigen-presenting cell, looking for an antigen stuck to the MHC molecule that could be recognized by the T cell receptor and might represent a pathogen. In this healthy mouse, it doesn't find anything. So you can see in the middle left panel that process is being withdrawn. And the T cell then, in the middle panel, goes over to another antigen-presenting cell, makes another process, and scans the surface of that cell. In the lower left, you can see that that process is being withdrawn. The T cell then becomes round again and leaves the picture. All of this interaction happens in about 2 seconds. So it's remarkable that Pia could provide this bunch of stills, which would allow us to see the process of immune surveillance of the nervous system as it occurs in real time in a living mouse. So the next cartoon summarizes our hypothesis about immune surveillance of the central nervous system. Immune surveillance is carried out by central memory CD4 cells in the spinal fluid. That's point number 1. On the right-hand upper panel, point number 2, these memory T cells enter the CSF across the choroid plexus and a little bit across the meningeal veins where the CSF is being produced. And then point number 3, how do these memory T cells get out of the CSF and notify the immune system if there's a problem? They exit at the base of the forebrain across the cribriform plate, that very thin bone at the base of the skull that the frontal lobes sit on, and they percolate out along with spinal fluid through the channels that the olfactory nerve roots use to get in and synapse in the olfactory bulb. So the T cells percolate out into the nasal mucosa, and in the nasal mucosa they find an afferent lymphatic, which allows them to get into a deep cervical lymph node. At the time that we initially made this proposal, it was just a sort of a guess. It made sense to us. And then subsequently, as you see in the lower right-hand panel off to the side, Beckman and colleagues actually found evidence to show that our guess was correct and published that in year 2006. So these T cells use the trafficking pattern that is characteristic of central memory T cells, but they do so in a way that's particularly adapted to monitor the central nervous system. They start in the blood, in the stromal vessels of the choroid plexus. They leave the blood, cross the epithelium of the choroid plexus, and enter the spinal fluid. So they go from blood into a tissue fluid rather than into the tissue itself. Within that tissue fluid, these cells encounter lots of antigen-presenting cells in the form of the meningeal macrophages in the subarachnoid space, and those macrophages spend their time sampling the CSF, which is in equilibration with the interstitial fluid of the brain. So they get a complete sample of the interstitial fluid of the brain. So the T cells go from blood to the tissue fluid, the spinal fluid. Then they go back out the cribriform plate into a lymph node from the lymphatics in the nasal mucosa, and then finally from the lymph node in an efferent lymphatic, they go into the lymph, the thoracic duct, and back into the blood. So it's exactly the pattern that you see in any tissue for central memory T cells: blood, tissue, lymph node, blood. This is quite different, as you see on the next slide, from using the blood-brain barrier vessels to get into the brain. The top panel in the middle here shows the process of immune surveillance. It's carried out by central memory T cells that take the route from the stromal vessels of the choroid plexus across the epithelium and into the CSF. This is part of normal physiology, and it's happening within us and all healthy people all the time. The other process in the lower panel is the immune effector response. This is always associated with pathology. It involves activation of the blood-brain barrier vasculature. That vasculature interacts with effector memory T cells that are short-lived and with monocytes that are activated, and together they are able to enter the CNS across the disrupted blood-brain barrier. If they're going into the brain in the context of an infection, they're carrying out host defense. If they're going into the brain in the context of autoimmune inflammation, as we see in MS, they're part of the pathology. But this entry across the blood-brain barrier into the brain parenchyma is never normal and does not occur in the healthy brain. Now, how does this work at a detailed molecular level, and how can we use this knowledge to understand disease and its treatment? Whenever a cell traffics out of the blood, it uses a number of steps by which the leukocyte, which is in blue and is round, interacts with the endothelium, which is kind of purpley and sort of rectangular. And these interactions are called rolling, the initial interaction, which is very tenuous and reversible, activation, which is in the middle, adhesion, by which the leukocyte stops on the endothelium and sticks to the endothelium even though the blood is still flowing, and then finally diapedesis, by which the leukocyte squeezes between two endothelial cells and enters the tissue space. The usefulness of identifying these various steps in the interaction between the leukocyte and the endothelial cell was that it allowed for the identification of specific molecules that carry out each one of these steps: selectins for rolling, chemokines for activation, integrins for adhesion, and then finally again chemokines for migration from the stuck adhesive state through the endothelium and into the tissue. The reason that this interaction is able to carry out so many, or able to direct and orchestrate so many different types of processes, is shown on the next slide. So if you think of it, leukocytes spend all their time whizzing around in the blood. We have many different kinds of leukocytes. As you know, we have several different types of T-lymphocytes, of B-lymphocytes, monocytes, basophils, eosinophils, neutrophils, and so forth. And these leave the vasculature under all sorts of different circumstances. They may be trafficking into an inflamed tissue to fend off a pathogen. They may be leaving the blood simply to go into the spleen or into a lymph node as part of normal immune activity. They may be under developmental circumstances colonizing a tissue like the skin or the gut. So there are dozens of different circumstances by which leukocytes leave the blood and enter a tissue. And the question comes, how do the cells know what to do? And the answer is they use the same general paradigm every time, but they use different molecules depending on the circumstances. So of the selectins, which mediate the rolling, there are a few more than 5. Of the chemokines, which mediate activation, there are about 50. And of the integrins, which mediate arrest, again, there are about 5. So given that you only need one selectin, one chemokine, and one integrin each time a cell leaves the blood, you can see there's a lot of different combinations which can help specify which cell is leaving the blood under which circumstances for which purpose. And this now comes around to help us understand neurological disease. The drug that's used to treat multiple sclerosis that most directly affects leukocyte trafficking is called natalizumab. It's a monoclonal antibody which binds to and inactivates alpha-4 integrins. All the integrins have an alpha and a beta chain, and one of the flavors of alpha chain that is typical of memory lymphocytes is the alpha-4 integrin chain. So natalizumab blocks the alpha-4 integrin chain, and if you stop one step of the process, you stop the whole thing. So it doesn't matter if the cell has already undergone rolling activation. If you block the integrin, it will not arrest and it will not leave the bloodstream. So blocking any point in this cascade of interactions is enough to block the whole thing. So how do these interactions work? On the next slide, you can see how cells make up their mind to leave the bloodstream. What we're showing here is a high endothelial venule. You can see that label at the bottom on the left, HEV. So these are cells thinking about going into a lymph node. On the left, you see a memory cell, and the memory cell basically will see almost nothing on this endothelium of interest to it, so it just passes right through. And in this case, this would be an effector memory cell, not a central memory cell. Effector memory cells do not enter lymph nodes because they're highly activated and would cause a lot of trouble in the lymph node simply by stirring up all the cells that are already there. In the middle, you see a PMN, a neutrophil, polymorphonuclear leukocyte, and that cell has one molecule on its surface, the selectin, that allows it to engage in rolling along the HEV endothelium. So it rolls along and it's looking for an activation signal, which would be the next step, but it doesn't find anything. So you see a yes for the rolling step and a no for the activation step, and so it just ends up rolling right out of the picture. It will never arrest. Only the naive cell of these three is going to say, yes, I have the right selectins, yes, I have the right receptor for the chemokines that are displayed on this endothelium, and yes, I have the right integrins to interact with the integrin receptors, called CAMs, on this endothelium, so I can roll, I can receive the activation signal, I can arrest, and at that point I will then leave the vasculature and go into the lymph node, the process called homing. So once again, immune surveillance uses specific molecules to get cells out of the stromal vessels across the epithelium and into the CSF. So for CD3 lymphocytes, that would be T-selectin for the rolling, and for Th17 CD3 lymphocytes, the particular chemokine receptor would be CCR6, and it would use alpha-4 beta-1 integrin. And the alpha-4 integrin then can be blocked by natalizumab. So people receiving natalizumab have a reduced number of spinal fluid T cells. On the lower panel, you see some of the molecules that are used by the effector cells, the effector lymphocytes and activated monocytes, which interact with the activated blood-brain barrier endothelium to go into a site of pathology. In the case of the CD3 T cell, we know that alpha-4 beta-1 integrin is involved, just as it is for the spinal fluid T cells. In the case of monocytes, we know that the chemokine receptor CCR2 is important. But as you see, we can't fill in the selectins, we can't fill in some of the chemokines, we can't always fill in the integrins because we simply don't know all the details for this type of trafficking. So let's try to integrate this idea with how these various drugs for treating MS actually work. We talked already about natalizumab. Let's take a closer look now at what happens when T cells come in and out of lymph nodes. On the next slide, again, we're looking at adaptive immunity, and we're looking at priming and response in the lymph node. So on the left, in the upper left-hand corner, you see the word blood, and in this case, the blood is flowing through a high endothelial venule. So T cells can enter, as we said, either naive or memory T cells can enter that lymph node and look for antigen. When they do so, they have a receptor on the surface called S1P1. They don't use that receptor to get into the lymph node, but if they don't find anything, or if they do find something and need to leave the lymph node to go fight off a pathogen, in either case, they need to use that S1P1 receptor to exit the lymph node. So this is shown as the kind of lightning bolts right in the middle right-hand side of the cartoon. So they use the S1P1 receptor to leave the lymph node. The drug fingolimod, which is used for treating multiple sclerosis, blocks the interaction of the S1P1 receptor with its ligand, S1P, sphingosine 1-phosphate. And therefore, both naive and memory T cells are trapped within the lymph node, and they can't leave, and they're not available to go into the brain and cause MS attacks. how does interferon beta come into the picture? So it turns out that a molecule called CD69, a surface activation indicator, is turned on by interferon beta, and CD69 acts in a sense like fingolimod. When CD69 is inserted into the membrane of a T cell, it can migrate laterally through the membrane and bind to S1P1 and block its function, so that when people get interferon beta for MS, a certain number of their T cells are trapped within lymph nodes, exactly as we see with fingolimod. And those cells are not available to enter the brain and cause MS attacks. So in essence, we have 3 different drugs for MS, all of which target leukocyte trafficking in different ways. Natalizumab keeps cells from leaving the vasculature. Fingolimod traps cells in lymph nodes by one mean of blocking S1P1, and interferon beta traps lymphocytes in lymph nodes through a different means of blocking S1P1, and they all work. All right, now let's take all this information and get back to our patient. Remember, our patient has new MRI lesions after starting interferon beta for her relapsing remitting MS. So we have to wonder, what's causing this breakthrough disease? This is not a good thing that she has new lesions. In the context of treatment with interferon beta and other first-line drugs, new MRI lesions carries a prognostic indication that over time she will not do well with her MS and is at some risk for developing disability. So what is causing the breakthrough disease? We need to figure this out and deal with it. It may be neutralizing antibodies. She's been injecting interferon, and depending on the interferon used and the individual person, she may have a risk of neutralizing antibodies to interferon that is anywhere between 5 and 25%. So that's a consideration. Did we make the wrong choice of medication? That's another possibility. About 20% of people with relapsing remitting MS on interferon beta will show MRI or clinical breakthrough without any neutralizing antibodies. And these people have a poor prognosis on interferon beta. They need to be considered for switching to a second-line agent. So what are the second-line agents that we have? DMA in this context means disease-modifying agent, drugs used for treating MS. We can consider natalizumab, the alpha-4 integrin antibodies, fingolimod, the S1P1 receptor inhibitor. In the past, mitoxantrone, a cytotoxic immunosuppressive, was considered. These days, the side effects of mitoxantrone are such that it is quite rarely used for MS because the alternative drugs are superior. So mitoxantrone is no longer used in most clinics, particularly in the United States. So what are we going to do about decision making? Even back in 2012, we would not have used mitoxantrone. It's inappropriate because of toxicity. And remember, this woman's 32 and wants to start a family. Mitoxantrone will knock down fertility, so we're going to throw that one out. Natalizumab can be highly effective in this context, but its use is complicated by a rare but serious infection called progressive multifocal leukoencephalopathy. In order to carry out appropriate risk mitigation, we have to make certain that this patient has not received cytotoxic immunosuppressives at any time previous for any other medical condition. We know she hasn't because we said her past medical history was essentially no serious illness. But we can measure antibodies to the virus that causes PML, JC virus antibodies, and if those antibody tests show no antibodies to JC virus, then she hasn't been exposed to the virus and is at very low risk of developing PML during treatment with natalizumab. So that would move natalizumab up in the chain of consideration. Fingolimod is also effective in this context. It can be complicated by disseminated zoster, so it is considered appropriate for risk mitigation in using fingolimod to measure zoster antibodies, VZV antibodies. If she does not show evidence of adequate antibody-mediated protection against zoster, then it's appropriate to consider immunizing her with chickenpox virus vaccine so that you can protect her against disseminated zoster. So that wraps up our story for multiple sclerosis. I said that at the very end we want to consider T-cell homing in other diseases. So in the cartoon that I'm showing you, a naive T-cell, and there are some indications under the title there of what molecules are on the surface of that cell, and it's shown by a white ball. It gets stimulated by antigen, and in this case, if the stimulation by antigen occurs in a mesenteric lymph node, that lymph node will also be full of retinoic acid derived from vitamin A, and that signal, the retinoic acid, will tell the T cell not only this is your antigen, find the pathogen and fight it, but it will also say this antigen came from the intestine. So it programs the T cell with surface markers, homing markers, that are called gut homing markers, and it will go right back to the gut and find the pathogen. Alternatively, on the left-hand side of the slide, you see that in green, this is a peripheral lymph node dendritic cell, and it has presented antigen in a skin-specific lymph node. And that lymph node is going to be full of dihydroxy vitamin D. Remember, vitamin D is in part converted to its active form in the skin through the action of UVB light. And when a T cell is activated by antigen in a lymph node that's full of dihydroxy vitamin D, it will acquire a bunch of surface markers that cause it to migrate back to the skin. So it will become a skin homing T cell. And since the gut homing and the skin homing signatures involve specific molecules, it may be worth considering whether we could treat inflammatory diseases of the skin that involve T cells, such as psoriasis, by blocking skin homing, and inflammatory diseases of the gut that involve T cells, and this would be inflammatory bowel diseases, by blocking gut homing. So not just multiple sclerosis, but other major inflammatory diseases of humans could be treated in this way. The right-hand panel now simply shows this progressive scheme of differentiation by which a naive T cell is activated by antigen in contexts which either direct it to the skin, the gut, or to a variety of other tissues. So in conclusion, T-cell trafficking is central for immune responses. It is blocking T-cell trafficking is the best validated treatment strategy for multiple sclerosis. T-cell trafficking is mechanistically decipherable. By that I mean we can identify the molecules that get T cells to the various places in the body where they go. And when we know those molecules, we realize that trafficking to different tissues is different, and that we can block migration to one tissue without damaging the ability of T cells to carry out their missions in other tissues. And so, in conclusion, it's reasonable to say that it's appropriate to target T-cell trafficking to treat inflammatory disease. The specific targets and complications obviously will differ among varied diseases. That's the end of my presentation. Thank you, and take care.